Okay, so we're ready for chapter 2 in our exploration of the tobacco mosaic virus. So let's recall where we are. Uh, People have discovered this thing called a virus, which has these unusual properties. It appears to be totally inert and has a shelf life. It can just sit there and hang out on a shelf forever. But when you inject it into the appropriate host, it comes to life. It begins to reproduce itself. And this has tremendous significance at the time. So this period of time, 1900 to 1930 is where we're talking about. This has tremendous significance because over that 30-year period, science went through an amazing transformation. In 1900, physicists pretty much believed they knew everything there was to know. They were actually saying, we're close to having an answer to everything. And in a period of about 5 years, they discovered they knew almost nothing. Along comes Einstein and theory of relativity and general relativity, and then all of a sudden we discover these particles that have these wave-like properties and these particle-like properties, and we discover the laws of quantum mechanics, and all of a sudden everything goes out the window, and we discover we know nothing at all. At the same time, chemistry, which had been pretty much an empirical science, where we pour a little of this with a little of that and you get this, and that's how people proceeded. They just played with these chemical reactions and they had no real reason for them. As a matter of fact, they thought atoms were held together by hooks and eyes. So they'd come up and we have a molecule, we have this hook on one and an eye on another, and they'd hook together and form molecules. But by 1923, 24, 25, 26, this was just, no, this isn't the way things work at all. They all work because of the allowed energy states of electrons, which are given to us by quantum mechanics. So all of a sudden, in this period of time, everything just blew up. And that brought a very interesting question to mind. There were groups of people who said, life can be explained using chemistry and physics. That is, life obeys the rules of chemistry and physics. On the other hand, other people said, no, natural laws now do not allow us to understand life. And there are other natural laws, or maybe it's just not understandable at all. Maybe it's something that we're not allowed to know anything about. And in the virus, it seemed that this was the bridge. This was the bridge, because what is that property of life that we really want to know about? And the property of life is, of course, reproduction. Life makes life. And whereas we can build machines that do everything that living things do, we can't make machines which make themselves, or we couldn't at that time. And there's still a question whether we can do it now. But so the great interesting property, the great interesting story of viruses were these things that seem to fall to provide a link between, if we could understand viruses that live in this no man's land between living and dead, between animate and inanimate, maybe we can put together what's going on. So there are different schools. And one of the great schools that said we can understand what's going on in schools of thought, that is, one of the great schools of thought which said we can understand life based on the principles of physics and chemistry was the Rockefeller Institute at Princeton. And all the people there were basically committed to this idea that things were not beyond understanding and the laws of physics and chemistry were sufficient. And so they were involved in this search for the structure associated with reproduction, with being able to make copies of itself. And their preferred candidate were enzymes. And enzymes are molecules that catalyze biological chemical reactions. Now, enzymes fall into a class of things called proteins. And proteins are nothing more than linear polymers made up of 22 naturally occurring amino acids. So they're just a polymer where I have amino acid 1 here, amino acid 2 here, bump, 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 bump. And I just hook these things together, these 22 different possibilities, in a huge, an infinite number of possible ways. And so I've got this huge possibility for the number of proteins we can generate. Now, natural proteins fall into kind of two broad classes. One of them is structural proteins, and you're familiar with those. Hair is a structural protein. Fingernails are a structural protein. On the other hand, enzymes are the catalysts that lower the energy of reactions and makes reactions go faster. For example, in your digestive tract, you have enzymes in your system that break down the food into useful components. Those are digestive enzymes. You can't eat wood because you don't have the enzyme that breaks cellulose down into its sugar components. On the other hand, termites have exactly that enzyme in their system, so they can consume wood. So basically, enzymes are the moving, humming parts of a cell's machinery. And it's not surprising that enzymes were seen as the key to understanding life, because they are, they're what's going on, they're what's moving, there's what's making things happen. When you walk in there, when you go into a cell, you see the enzymes, you go, whoa, that must be where it's all going on. So also, in about this time, in about the 1930s, enzymes were discovered that were called autocatalytic. That is, they could catalyze their own synthesis. You take a few molecules along with the precursors for this enzyme, and you start the reaction going, and those few enzymes lower the energy to make others. And as those come along, they lower the energy to make others. And so in a very short period of time, you're making lots of these enzymes. So those are called autocatalytic, in that they can reduce the energy barrier to making themselves. Well, this seemed a lot like reproduction. So these systems, autocatalytic enzymes, seemed like the ideal candidate to study in order to get at this idea of living life. What's the structure that gives rise to the property of reproduction? And many people were guessing that viruses were a very special autocatalytic enzyme that bridged the animate and the inanimate world, and that viruses might be the transmitter of genetic information. That is, genes and viruses may have been closely linked. Genes had been known since the time of Mendel to be somehow transmitted from father and mother to daughter, etc. And so they were the transmitters of that. All right. And further, if you're going to study viruses and want to understand this property, you've got to get at their structure. So here we have people going, there's this real interest, the broad interest of saying, can we understand the structure of viruses? And of course, as we've mentioned, the way to do that at the time was through X-ray crystallography. Now, this brings us to 1936, when after several years, this young scientist, Wendell Stanley, I think he was about 28, 29, somewhere in that ballpark. So he was a really young guy. And he'd been working at the Rockefeller Institute and using the techniques that many people there used to crystallize enzymes, he succeeded in crystallizing the tobacco mosaic virus. And Stanley published his results in a prestigious journal, Science. And in that journal, he concluded that the tobacco mosaic virus is an autocatalytic protein that requires a living cell for multiplication. So see, what he's saying is the virus is in fact a protein. It's an enzyme. And somehow that enzyme gets into a cell and it autocatalytically reproduces itself. So this is huge. And with his ability now to isolate, to grow it as a crystal, to isolate it, all sorts of things became possible. So you could now do chemical assays on this and determine what it was made of, what its shape was, its molecular weight, how it compared to other viruses. And so all sorts of things are going on right now as we develop. Actually, this stimulated the production of many new techniques to isolate viruses of all sorts, not just the tobacco mosaic virus. But we came up with new methods called ultracentrifugation and gel electrophoresis, and all these new ways to look at viruses. And as we're looking at viruses now, we can compare them. We can compare the virus, the TMV virus, to other viruses, including those which affect human health. So the scientific world was absolutely rocked by this accomplishment. And part of the reason was the public was so struck by this because a crystal seems to be like a mineral, like a salt. And yet, on encountering a cell, this crystal sprang to life. It was remarkable, and it began to reproduce itself through catalytic mechanisms. So this was cool. The New York Times actually ran a headline, I think in 1936, could have been 1937, that says, "Crystals isolated at Princeton believed unseen disease virus." The Pittsburgh Press implied that the viral disease could be caused by a dead chemical like salt or iron ore. And then they went on to say this is the final missing link between dead of the chemical world and living of the biological world, and the earliest source of what we call life. So you can see everybody's swept up in this. This is just incredible. And of course, Dr. Stanley is getting huge amounts of press for all of this. Influential scientists at the time pointed at this and said, virus proteins are naked genes. The virus protein actually carries the gene. And somehow they pictured that like chromosomes or big linkages of these things like viruses could now autocatalytically reproduce themselves. So everything's thought to be proteins and enzymes. Now, the problem is that this whole idea is just plain wrong. The protein portion of the virus is a structural protein. It's simply the container for its genetic contents. And we know this now, but people did not know that then. But as always in science, other researchers began to take a closer look because of all these techniques that had come along, and those techniques came along because of Stanley's effort. That's important. As he developed this idea, other techniques came along so we could begin to look at viruses. And using these rapidly developing techniques, the tobacco mosaic virus was found not to be a pure protein. It contained 6% of what's called nucleic acid. That was interesting to people. And this nucleic acid, they were able to show, actually made this virus active, made it so it could infect a host. So they had preliminary suggestions of that. And as they isolated other viruses, they also discovered that all of them contained, as a minor component, nucleic acids. On the other hand, as they began to isolate more and more enzymes and crystallize those and separate those out, they didn't find a single enzyme that contained nucleic acids, not a single one. So the idea that viruses were autocatalytic proteins wasn't quite right. Now, Stanley at the time called them nucleoproteins. So he said, oh yeah, they're a little different, but they're nucleoproteins. So that's what was pushing at the time. Now, this is about 1940. And at this point, we're going to take a 5-year hiatus because all work on viruses and enzymes, at least most of it, just ground to an absolute halt as World War II diverted scientists into other areas of study related to the war effort. So there's this 5-year hunk in there where nothing happens. But immediately after the war, 1946, Stanley received the Nobel Prize for Chemistry. And in the introduction at the Nobel ceremonies, they said, Dr. Wendell Stanley, we owe you one of the most striking discoveries in modern chemistry and biology, the demonstration of the fact that viruses can be crystallized in the same way as many proteins and enzymes, and that a virus is actually a protein. So they're saying the structure and the properties of the virus comes from its protein structure. Okay. Again, he's got the Nobel Prize for this, and it's wrong. But the questions that came up before the war, where they were seeing the nucleic acids, those remain. Scientific questions don't go away just because of a war. And with the end of the war, scientists began to revisit these things. And within just a very few years, it will lead to the most amazing, probably one of the, if not the greatest scientific discovery of the 20th century. And we'll pick up there on the next one. Have a good day. See you in just a few minutes in lecture. I leave you with this picture of the tobacco mosaic virus.